Hello, my dear students. Today, let us discuss about the topic structures and functions of different components in the digestive system to digestive glands. In a human body, several glands are involved in the digestion process. Digestive glands are the organs through which food passes during its processing and contribute juices necessary for the digestive process. The digestive glands include salivary glands, gastric glands, liver, pancreas, and intestinal glands. Number 1. Salivary glands. The salivary glands located under the tongue and in the back of the mouth discharge their secretion into the oral cavity. There are 3 pairs of salivary glands, namely parotid, sublingual, and submaxillary or submandibular. Number 1. Parotid glands. These are the largest salivary glands. They lie on the sides of the face, just below and in front of the ears. The parotid ducts, which is also known as tension ducts, open into the vestibules opposite the upper second molar teeth. Viral infection of the parotid glands causing swelling and pain is the disease called mumps. Next, sublingual glands. These lie under the front part of the tongue. The sublingual ducts, also known as ducts of Rivinus, also open under the tongue. Next one is submaxillary glands. These lie at the angles of the lower jaw. The submaxillary ducts, also known as Wharton ducts, open under the tongue. Next comes saliva. The salivary glands secrete a viscous fluid known as saliva. It contains water, salts, mucin, and an enzyme, salivary amylase or ptyalin. Its pH is nearly neutral, being 6.7. The salivary glands are under neural control. Salivation is activated by the sight, smell, idea, and talk of the food and by the presence of food in the buccal cavity. About 99.5% of saliva is water. It contains 2 important enzymes, amylase and lysozyme. Amylase is a starch digesting enzyme, breaking starch into maltose and triose. Lysozyme causes lysis of several common bacteria that may be present in the mouth. Mucin in saliva helps to lubricate the food for swallowing. Next comes functions of saliva. Number 1. It moistens and lubricates the buccal mucosa, tongue, and lips, thus making speech possible. Next, it also moistens food and changes it to a semi-solid mass for easy swallowing. Next, saliva washes mouth and tongue clear of cellular and food debris. In fever, the salivary secretion is reduced and tongue becomes dirty or coated. Next, It neutralizes acidity in the buccal cavity. Next, its enzymes helps in digestion. Next comes to gastric glands. Gastric glands are simple, branched and tubular and million in numbers. They are of three types: the cardiac glands, which are located in cardiac part of the stomach; the pyloric glands located in pyloric part of stomach; and the fundic glands that comprise of many types of cells located in the epithelium. Next comes functions of cardiac glands. They secrete mucin and very little pepsinogen. Next, pyloric glands. Their secretion is rich in mucin and does not contain hydrochloric acid. Next, fundic glands. They are also called oxyntic glands. They secrete hydrochloric acid, pepsinogen and soluble mucin, acidic secretions. Types of cells present in the epithelium of the gastric glands are peptic cells or sheath or zymogenic or zymogen cells, oxyntic or parietal cells, and mucous cells or goblet cells. Sheath cells are usually basal in location and secrete gastric digestive enzymes as proenzymes, pepsinogen and prorenin. Oxyntic or parietal cells are large and most numerous on the side walls of the gastric glands. They are called parietal cells as they lie against the basement membrane. They secrete hydrochloric acid and Castile intrinsic factor. Mucous cells or goblet cells are present between other types of cells and secrete mucus. Mucus is a glycoprotein and help to neutralize the acid in stomach and protects stomach wall against hydrochloric acid action and protein digestive enzyme. The epithelium of the gastric glands also has two types of cells, namely endocrine cells and stem cells. Endocrine cells are usually present in the basal part of the gastric glands. These are argentaphil cells and G cells. Argentaphil cells produce serotonin, somatostatin and histamine. G cells are present in the pyloric region and secrete and store the hormone gastrin. Stem cells are undifferentiated cells that are also present in the epithelium of the gastric glands. They multiply and replace other cells. They increase in number when the gastric epithelium is damaged and play an important role in healing. Next comes gastric juice. The secretions of the cells of gastric glands form gastric juice with pH 2 to 3.7. About 2000 to 3000 ml of gastric juice is secreted per day. The gastric juice contains two proenzymes, namely propepsin and prorenin, with enzyme gastric lipase, mucus and hydrochloric acid for the digestion of proteins and fats. Next comes to liver. Liver is the largest digestive gland of the body, which is brown in color. It lies in the upper right side of the abdominal cavity, just below the diaphragm. It is heavier in males than females. In males, it is generally weighed 1.4 to 1.8 kg and 1.2 to 1.4 kg in females. The liver is divided into two main lobes, right and left lobes, separated by the falciform ligament. The falciform is a membrane that is continuous with the peritoneum. The right lobe of the liver is further differentiated into three lobes, such as right lobe proper, a quadrate lobe, and a caudate lobe on the posterior surface. Internally, the structural and the functional units of liver are the hepatic lobules containing hepatic cells arranged in the form of cords. Each lobule is covered by a thin connective tissue sheet known as the Gleason capsules. The Gleason capsule is the characteristic feature of the mammalian liver. The mammalian liver also contains Kupffer cells that are phagocytic cells, and it burns out WBCs, RBCs, and bacteria. Next comes to functions of liver. Number 1, production of bile. Bile is stored in the gallbladder. About 500 to 1000 ml of bile is secreted by liver in a day. Number 2, deamination. It is a process by which the amino group is removed from the amino acids, resulting in the production of ammonia, which is converted into urea. Next function is excretion. Liver synthesizes urea with the help of ammonia and carbon dioxide. Next function is glycogenesis. It is the conversion of the excess of glucose into glycogen by liver cells with the help of insulin secreted by the pancreas. Next function is glycogenolysis. Formation of glucose or glycogen from non-carbohydrate sources such as amino acids, fatty acids, glycerol, etc. The next function is lipogenesis. Conversion of excess of glucose and amino acids into fats. Next function is gluconeogenesis. Formation of glucose or glycogen from non-carbohydrate sources such as amino acids, fatty acids, glycerols, etc. Next function is detoxification. Liver converts toxic substances into harmless substances. Next function is hemopoiesis. It is the process for the formation of the blood. Next function is synthesis of blood protein. Next one is synthesis of heparin. Next one is lymph formation. Next, synthesis of vitamin A. Next, secretion of enzymes. Next, destruction of red blood corpuscles. Next, phagocytosis. Next, osmoregulation. Next, production of heat. Next, storage. Next comes biliary system. The gallbladder and the ducts that carry bile and other digestive enzymes from the liver, gallbladder, and pancreas to the small intestine are called biliary system. Next comes bile. The liver cells secrete bile via the hepatic duct to the gallbladder for storage and hence on demand via the common bile duct to an opening near the pancreatic duct in the duodenum. Bile is an alkaline, viscous, greenish yellow fluid. About 500 to 1000 ml of bile is secreted by liver in a day. It is released into the duodenum on cholecystokinin CCK stimulation. Bile contains bile salts, bile pigments, cholesterols and phospholipid. Next comes functions of bile. Number 1, neutralization of hydrochloric acid. Number 2, emulsification. Number 3, absorption of fat and fat-soluble vitamins. Number 4, excretion. 5, prevention of decomposition. 6, stimulation of peristalsis. And number 7, activation of lipase. Next comes to pancreas. Pancreas is a soft lobulated grayish pink gland which weighs about 60 gram. It is about 2.5 cm wide and 12 to 15 cm long, located posterior to the stomach in the abdominal cavity. The pancreas comprises the head, body and tail. The head lies in the C-shaped curve of the duodenum, the body behind the stomach, and the tail reaches the spleen lying in front of the left kidney. It consists of two parts, number 1, exocrine part and number 2, endocrine part. First one, exocrine part. The exocrine part of the pancreas consists of rounded lobules or acini that secrete an alkaline pancreatic juice with pH 8.4. About 500 to 800 ml of pancreatic juice is secreted per day. The pancreatic juice is carried by the main pancreatic duct into the duodenum through the hepatopancreatic ampulla. The pancreatic juice helps in the digestion of starch, proteins, nucleic acids and fats. Therefore, pancreatic juice is also called complete digestive juice. Next one, endocrine part. The endocrine part of the pancreas consists of islets of Langerhans. The human pancreas has about 1 million islets. They are most numerous in the tail of the pancreas. Each islets of Langerhans consists of 3 types of cells which secrete hormones to be passed into the circulating blood. The alpha cells secreting hormone glucagon, beta cells secreting hormone insulin, and delta cells producing hormone gastrin and somatostatin. Next comes intestinal glands. The intestinal glands are numerous and microscopic. They lie in the wall of the small intestine. They are of 2 types, Krebs of Leverkusen and Brunner's gland. Krebs of Leverkusen are simply tubular glands and occur throughout the small intestine between the villi. They secrete enzymes and mucus. They have 2 types of cells that are Paneth cells and argentaphil cells. pannate cells secreting lysozyme, is antibacterial in function, and argentaphil cells play a role in stimulating peristaltic activity of the intestine. Brunner's glands are branched tubular glands and are confined to the duodenum. They secrete alkaline watery fluid with little enzyme and mucus. From the above discussion, it is concluded that the human digestive glands include salivary glands, gastric glands, liver, pancreas, and intestinal glands. The gastric glands are present in the mucosa of the stomach. These are of three types: cardiac glands, pyloric glands, and fundic glands. Liver is the largest digestive gland and consists of a large right lobe, a small left lobe, and two small lobes called quadrate lobe and caudate lobe behind the main lobes. Liver is helpful in blood clotting, formation of glycogen, regulation of blood sugar, as reserve food, synthesis of vitamin A, and storage of some minerals. These are some of the structures and functions of the digestive glands. Thank you.